Hello, everyone, and welcome to Arthritis Talks. I'm Sean Bevan, the Chief Science Officer at the Arthritis Society. Thank you for joining us tonight. While we've come together for this event from many different places, I would like to acknowledge the land on which I am located on, which is also where our Toronto office stands, and this is the traditional territories of the Mississauga of the Credit First Nations, Anishinaabeg, Huron-Wendat, and Haudenosaunee Indigenous people. The territory was a subject of the Dish with One Spoon Wampum Belt Covenant, an agreement between the Haudenosaunee and Anishinaabeg and allied nations to peacefully share and care for the resources around the Great Lakes. Today, this meeting place is home to many First Nations, Inuit, and Métis peoples, and I am grateful to have the opportunity to live and work on this land. Today, we are very fortunate to be joined by rheumatologist Dr. Tom Appleton, who is here to answer many of the common questions we receive about biosimilar medications. As provincial governments are moving to or considering transitioning to biosimilars, we are receiving more and more questions about them. We understand that biosimilar medications are complex and can be confusing, so we do hope that tonight will help to reduce some of that confusion. Before we get started, a few logistics. This webinar is best viewed on a laptop or desktop computer. If you have technical difficulties, please email arthritistalks@arthritis.ca for assistance. If you have a question for our presenter, you can submit it through the Q&A, the button at the bottom middle of the screen. We will try to get to as many questions as possible during the hour that we have together. You can also click on the chat icon in the bottom of the screen to access the chat and connect with other participants, as well as the Arthritis Society's chat moderator. If you'd like to close the chat completely, just click the red X icon to close out of the window. We're also pleased to continue to provide captioning of our webinars to accommodate the diverse needs of our audience, and you will see that running along the bottom of your page. So as usual, many questions we received followed similar themes, so we will address those first before going into the live Q&A at the end of the session. Before we get started, I want to thank our event partners, Pfizer, United Way Winnipeg, and our other partners for their financial support of our Arthritis Talk series. Now, let's get started, and first, a very warm welcome to Dr. Appleton. Dr. Appleton, let's please get started with the basics. So first and foremost, what are biosimilars?
Thanks so much for that question, Sean, and thank you as well to you and the rest of the Arthritis Society for putting on these sessions. The questions that are coming up and we're going to try to address for you tonight, as well as probably several others in the live Q&A, are questions that I get in clinic every day and that a lot of you are asking various sources, your different specialists, potentially primary healthcare providers, asking Arthritis Society and other sources for information on this. And I think it's really important that we try to get as much information out there as possible and as frequently as possible. So hopefully this is helpful. And I just want to say kudos to Arthritis Society for making this happen. To start off with your first question about what are biosimilars, I think one of the best places to start is, first of all, what are biologics? And this is actually a question that we got long before there were biosimilars, but continue to get at equal frequency. So what is a biologic medication? Most of us think of medications as those that are chemical based, so pill form kind of medications, and they're small molecules, whereas a biologic is much different than that. And I think it's important to put that in context with, is this a new thing or is this not a new thing at all? Well, it's not a new thing. We've actually been using biologics for a long time in Canada and certainly around the world. Biologics have been used specifically in rheumatology, though, to treat serious and life-threatening rheumatic diseases for more than 20 years. Actually, the first ones were approved in the late 1990s in the rheumatology space. And even before that, there are other areas of medicine that have been using biologics. So this is not at all a new technology, but it's a very important one. And I think it's really a landmark change that happened in the history of the rheumatology field when biologics were first introduced. They've completely changed the outlook for our patients. The most important thing has been the ability to control diseases that previously were not able to be well controlled. And certainly for a lot of patients, this has made a huge difference in their quality of life and their length of life. And biologics have really played a huge part in all of that. So let's go back to the distinction, first of all, between small molecules, which are more traditional or conventional medicines, and biologics. So small molecules are things like an aspirin, for example, a very small molecule that you take as a pill by mouth and you swallow it, it gets absorbed into the body. On the other hand, biologics are large proteins. And that means that you can't actually take them by mouth. They would be digested and they wouldn't work. So you have to inject them under the skin or take them through an intravenous route. And that's one of the key differences that patients really appreciate with biologic medicines. Because of their size and because of the fact that they work very much like an antibody, and some of them are in fact antibodies, they're very specific. And this is a special property of biologic medicines because it allows you to target the inflammatory protein that's causing some of the problems and remove it from the body or just inhibit it. Whereas small molecule inhibitors often have other effects on other targets. So biologics are often regarded as very clean medicines. They have very few off-target effects. In Canada, and especially many other places around the world, the way we use biologics is not as a first-line medicine. They're typically used after a number or at least one other small molecule conventional anti-rheumatic medicine is used. And many of you will be very acquainted with methotrexate, with things like hydroxychloroquine or sulfasalazine or leflunomide. These are conventional anti-rheumatic medicines that are used typically as first line and then followed as an advanced treatment with biologics. So biologics, again, came in at the end of the 1990s and completely transformed the way that we treat rheumatic diseases. So the way that they're made is from a live cell source. Initially, a cell is grown, it's cultured, and these cells will secrete the biologic proteins. The proteins can then be purified through columns and concentrated and ultimately synthesized into a biological drug product that can be dispensed and then, as I mentioned before, is given by injection under the skin. So this is how we make, or the manufacturing process for biologics. Just to give you a size comparison here. So I mentioned aspirin earlier. Aspirin is a very small molecule, which you can see on the left-hand side of the screen. And on the right, you can see a monoclonal antibody, which is a biologic type of medicine. And you can see that it is hundreds-fold larger in size and much more complex. And this is really important for the distinction between a biosimilar and a generic medicine. A generic medicine is very easy to copy. So aspirin can be copied exactly many different ways. But a monoclonal antibody, it's virtually impossible to make an identical copy. And that's why we call them biosimilars. So what is a biosimilar? It's just that, it's a biologic. We call it biosimilar because it is similar to something, and that something is the original version of the biologic that was made. We call those the originator biologics. They're allowed to be manufactured after the originator's patent runs out, and that's usually after 10, 12, 15, or maybe more years than that, depending on which jurisdiction you're in. And the biosimilars work in exactly the same way as the originator biologic. So they work on the same target, they have the same properties, and they're manufactured by the same process as the originator. So just to finish answering this first question, I wanted to give an example, and this example is familiar to many of you, which is adalimumab. Now, many of you will know adalimumab as the drug named Humira, but its drug name is actually adalimumab, and it has been approved and available in Canada since July 2006. And you can see that its distributor on the right is AbbVie. But since May of 2018, when the patent ran out for Humira, other companies were allowed to come in and manufacture adalimumab. And you can see that there are now 5 different biosimilar versions of adalimumab. Same drug, just has a different name. And you can see the different approval dates in 2018 and then several more in 2020, which came along with their corresponding distributors there on the right-hand side of the screen. So that's what biosimilars are. And Sean, I guess the context here is that biologics have completely changed what we do in rheumatology. And I wanted to know from your perspective at the Arthritis Society, what would you say the main impact has been since the advent of biologics in Canada on what you do at the Arthritis Society?
Yeah, that's a great question, Tom. I think that, biologics have really changed the treatment landscape, as you know, for many, many patients. So, thinking about people's quality of life quite differently, thinking about having really robust treatment options has been really revolutionary, I think, for patients. So now when it comes to the kind of patient supports that we offer, currently we are thinking a lot about how do we make sure, like doing this webinar, how do we make sure that patients have the right information to understand these new treatment options in order for them to have the best outcome. So thank you for all of that, Dr. Appleton. I think that this is an important question, and really, how do we know that biosimilars are in fact safe?
Well, this is one that comes up all the time. And, I think when biosimilars first came out, there was a tremendous amount, appropriately, of concern and really people wanting to make sure that what we were doing with the biosimilar manufacturing process was going to be at least as safe, if not even more safe than what has been previously happening for the last 2 decades with biologic production. And this is only natural, I think, that comes up. So this is a really important question. How do we know that they're safe? Well, we know because they're tested and they have to go through a rigorous process of approval in order to be available. And that includes Canada, but also includes almost all other jurisdictions where there are regulatory authorities around the world. In order for a biosimilar to be approved, it has to demonstrate that it has the same efficacy, and that means how well a drug works, to the originator biologic. So it's directly compared in studies to the originator biologic, at least for one of the major indications. So for us in rheumatology, one of the most common ones that is tested is for rheumatoid arthritis. If a biologic has been used to treat rheumatoid arthritis for many years and a biosimilar wants to come and be used to be treated for rheumatoid arthritis, it has to be treated for that. It has to be tested in multiple trials and shown to have similar efficacy and to have similar safety. So to date, and if I'm really thinking about this around the world, there have been more than 170, actually 178 at last count, more coming out all the time, of studies within rheumatology, in gastroenterology, dermatology, that demonstrate that there really is no clinically meaningful difference in the safety or the efficacy of biosimilars when you compare them directly to their originators. So that's quite reassuring. This is language that came from the European Union, and in the European Union, the EMA, or the European Medicines Agency, is the regulatory authority. For us in Canada, it's Health Canada, of course. In Europe, it's the EMA. And so the language that was put forth was that the range of variability for a biosimilar has to be the same that is allowed for batches of the reference medicine. So what do I mean by batches? Any manufacturing process, things are typically made in batches. You'll make a whole bunch of them at once, and then you'll start another batch, and on you go. And so that means that from batch to batch, there may be some subtle variance. And of course, there's lots of quality control and quality assurances on manufacture of automobiles, manufacture of other medicines, and it's exactly the same for biologics. And so that variability is well recognized. And so the European agency said many years ago that if we're going to approve biosimilars, it should be no more than the variance between the batches of the biologic originator that was originally approved. So this is achieved by maintaining a constant standard of robust manufacturing processes, quality control standards, and monitoring the batch-to-batch variability over time. And that continues even after approval. So just to give you an idea of what we mean when the regulatory authorities actually review biosimilars. So I've put Health Canada here at the top, and that's of course our regulatory authority here in Canada that approves all medicines that are used in healthcare. And you can see that the language is really quite similar across the board for the FDA, which is the middle one. So the FDA is the Food and Drug Administration in the United States, and the EMA, or European Medicines Agency, which I already mentioned at the bottom. They all have language that says that there must be demonstrated similarity to the reference biologic drug. And so that's really important for ensuring that consistency when we're talking about biosimilars, and especially transitioning from a biologic originator to a biosimilar. So, Sean, I think back to, when biologics first came out, and then biosimilars, and how that's really generated concern in the early days, to now, after several years of watching this unfold, after watching the science be published and reviewed, and the regulatory agencies review all of these sorts of things, really seen a shift in the focus to being, away from what's the major concern about safety to, how can we best use these sorts of medications. But I imagine at the Arthritis Society, there's also been some shifts over time. So what have you noticed as the main questions that you get, let's say, even through the AREP program at Arthritis Society about biosimilars?
Yeah, I think that one of the major shifts that we have had really is around, for us, as you just said, following the latest evidence and making sure that the information that we are providing is true to that. So I would say the Arthritis Society at large, we've been following this story for a long time, and trying to do our best to educate governments as they make these policy changes, to do so in the most informed ways. I would say that we've actually seen some significant changes over the last couple of years as we start to see provinces adopting these policies. And I would say that while understandably there's always lots of questions when people's medications are changing, I think that over time we're seeing a lot more comfort with biosimilars, which is really helpful, but I think also highlights the need for this kind of information. So I guess importantly, this is based on some of the questions that we have here, but what is the difference really between biosimilars and biologics?
Yeah, so this is definitely one that comes up, and I think it's core to the question of understanding what the role of biosimilars are. Biosimilars are not meant to be better than the originator. They're meant to be similar or even the same, at least when it comes to the safety and the efficacy. So in this infographic, which is actually produced by the Canadian Arthritis Patient Alliance, you can see that on the left, the biologic is in the turquoise green and the biosimilar is in the blue, and really talking about these different properties, how they stack up to one another. So similarities between biologic and biosimilar are the fact that they're both proteins, they're both grown and isolated from living cells. They're complex and expensive to make. That doesn't change just because it's a biosimilar. They're grown under strict manufacturing conditions. That's really key for any biological kind of process. And the cells themselves are programmed to make these specific proteins. And that's important because you can actually test that. You can validate that. It's not just a matter of getting it approved and then we forget about it. The ongoing monitoring that happens for biosimilars is exactly the same as the ongoing monitoring that had to happen for all of the biologics after they were initially approved. In terms of the results, it's the same protein, it works the same way. There are subtle differences in the chemical composition. And if you think back to that picture that I showed at the beginning of that massive antibody compared to the teeny little small molecule, there are going to be little tiny modifications around that protein, which could be detected by chemical assays. But the most important thing is that it doesn't mean that there's any difference in how well the biologic medicine works and how safe the biological medicine is. So whether you're talking about treating inflammatory arthritis like rheumatoid arthritis, ankylosing spondylitis, psoriatic arthritis, like I do and my colleagues do in clinic, or you're talking about treatment for anemia or breast cancer, these are all other areas where biologics are used. Health Canada requires that all the biosimilars have the same safety data, the same efficacy data, and the same post-marketing, meaning monitoring data. And that really the only difference comes in, there's a fewer studies that actually need to be done because we know that once it performs as well in one indication, it will perform as well in other indications. And that's part of the reason that these medications can be made more cost effectively, because you don't have to do as many large expensive studies. But nonetheless, they have the same safety and the same efficacy. So what are the differences? Well, they're kind of like two zebras with slightly different stripes, but they're basically the same thing. So it's a different version of the same biologic. It's produced by a different manufacturer. There may also be a different patient support program. And many of you who have used biologics will know about patient support programs. Even if you're not using it, you're probably still aware of it. The patient support program's main job is to ensure that you have access to the medicine and to make sure that it gets covered, whether that's by your private insurance provider or by the public provider in your province or territory. That's the primary role of the patient support program. They have other roles as well, which include helping get the medicine to you, helping to communicate back and forth between your physician's office and other health resources. But nonetheless, the main role is to help get you on and to support the medication. As a result, though, of the manufacturing process, these medicines are still fairly expensive, but able to be produced at a lower cost. And that's one of the main differences between biosimilars and biologics, is there's a lower cost to the health system by using a biosimilar compared to the original biologic drug. And we already talked about those chemical features being minor, but not associated with worse or different clinical efficacy or safety. So Sean, what is Arthritis Society doing these days to help patients understand biosimilars? Aside from tonight's session, of course.
Absolutely, tonight's session is critical to that. We also spent quite a bit of time building up our online resources, because we know that many people like to learn online to find the information when they want it. So we do have a very robust biosimilars hub on our website. So that would be a very key place, I would say, for people to go to learn more about what's happening across the country. Tom, we've already started to touch on this, but I think it would be helpful for people to hear more about it from you. We know that patients are transitioning from original biologic medications to biosimilars in many parts of the country. So could you elaborate and explain a little bit more about what that transition is that's occurring, and what does that transition typically look like for patients?
You bet. This is an important one. And actually, there are probably many people on the call tonight who have actually been through a transition process. And I think that's also an important resource. Everyone out there in the community who has been through this can speak to one another and help each other go through this. But I'll just outline it here, and then we'll probably have some more questions about it too in the live Q&A. I think, first of all, it's really important to understand the why. Why are we doing this? Why are we as a country, and why are we as different provinces across this country moving ahead with a transition process? And really the primary and almost only reason for doing this is the fact that they can be provided to patients at a lower price than the originator biologics. And this is exactly the same rationale for why generic versions of other medicines have been approved in the past. It's a cost savings, and that's really important for the healthcare system, for the sustainability of the healthcare system, and also to create opportunities to invest in other things that are important for patient care and improving patient outcomes. And so that's really the primary and, if not sole, motivator. So just to give you an idea, one of the main bodies in Canada that helps to establish or set what medication prices are like, or negotiate those in Canada, is something called the PMPRB, or the Patented Medicines Prices Review Board. Long name. They've estimated that the private and public drug plans in Canada could save between $332 million and $1.8 billion per year after the third year or so. So once you reach steady state after transitions have been implemented. So those are big dollar numbers in terms of savings for health systems. And because we are so proud of our public health care system in Canada, our systems, I should say, because we're a network of multiple provincially delivered healthcare systems, savings to our systems, which are really savings for our taxpayers, enable us to do more with the resources that we have. And that's really key. So the savings from transitions to biosimilars can and should be reinvested into other aspects of healthcare. There's really no clear medical reason from a safety or efficacy standpoint not to do this, not to transition. And I outlined that already. So if it's safe and it's effective, then why not save some money for the healthcare system? So what is a biosimilar transition? Well, it really just means transitioning or changing from the originator biologic that somebody has been taking for some time, and then switching to the biosimilar version of the same medicine instead. I also think it's important to point out what it is not. And at this point in time, we are not talking about interchangeability. So interchangeability means switching back and forth between biosimilar and originator, or between different versions of a biosimilar. So right now, it's essentially transitioning from the biologic originator to a single biosimilar version of a medication, but not switching back and forth between them. So that's what we mean by interchangeability. So that's different than the transition itself. There are two contexts where a transition might happen. So there might be a medical reason to transition, and this happens and has actually been happening in Ontario, where I practice, and many other jurisdictions, if not all, throughout Canada for several years, is starting out on a biosimilar. So essentially not going through the biologic originator in the first place, since these are now available, and many provinces have had that as a biosimilar first strategy. So in that case, there would be a medical reason to start a biologic, and then you would choose the biologic biosimilar and not the originator version. What we're really talking about here tonight, though, is non-medical transition. We're talking about transitioning not for a medical reason, but for the administrative reason. And that's when the public or the private drug plan requires a patient to transition from an originator biologic onto the biosimilar purely because it's less expensive. And just as an example, several processes are already happening in multiple provinces where transition programs have already been implemented. And there are examples of that with infliximab, with etanercept, with adalimumab. There are others with rituximab as well. So just to give you an idea of where this has been happening, you can see British Columbia was the first to implement their biosimilar transition program in 2019, and others have followed suit, including the Northwest Territories, Alberta, Quebec, and then most recently New Brunswick and Nova Scotia have all implemented their transition programs or are completing it right now. And other provinces are expected to implement theirs in the coming months. So, after this concept of biosimilar transitions was initially raised, the Ontario Rheumatology Association, the Canadian Rheumatology Association, and many other groups across Canada have put out position statements. And, in rheumatology, we have really emphasized in our position statements that we support biosimilar transition for the benefit of cost savings to taxpayers and to the healthcare system, but also that there should be key considerations. And those include making sure that there's no net increase in cost to any patient who is transitioned. That's a key principle. And the other principle, which I've already alluded to, is making sure that the savings from transitions to biosimilars are reinvested in healthcare. So I know that this is really important to us as healthcare providers, but I wondered if, Sean, you wanted to comment on the Arthritis Society biosimilar position as well.
Yeah, I think, on behalf of people living with arthritis, we see many of the same things, and much of what you've talked about with respect to the positions of the Canadian Rheumatology Association and Ontario Rheumatology Association, I think we're very aligned. I would just add a few things to what you said, though, around making sure that there's enough time for patients and physicians to make this transition period, making sure that patients have the right information to make sure that they're knowledgeable and empowered about what's happening. And also understanding, what's the longer term outcome of patients, to make sure that when we are introducing these in the real world, to make sure that everything that we expect to happen is happening as it should. So a few additions there that I think are very well aligned with other associations across the country.
Yeah, you're absolutely right. And those are really important as well. to acknowledge. I also wanted to point out that, sort of what you can expect if you're going through a biosimilar transition. If you're calling in from BC, you've probably already been through this, or Alberta. Others are going through it now. Others will go through it in the coming months or maybe year or so. But regardless of the reason for the transition, you can be assured that your rheumatologist or whoever your specialist is going to discuss this with you and is going to help you select a new biosimilar. You may end up being enrolled in a new patient support program for that biosimilar where it's applicable. Not everybody needs to be in a patient support program, but many will be. And I mentioned before that the patient support program is there to help obtain and maintain access to the medication and coverage for it. And you can also be assured that patients and their rheumatologists will be monitoring the efficacy and the safety after the transition occurs, just to make sure that everything is going well, as it should be, and we fully expect that it will be, and to just be reassured that that process will unfold.
So that's great, Tom. Thank you so much for all that expertise with us tonight. We will be getting into the live Q&A in just a moment, but before we do that, do you have any final thoughts or recommendations that you wanted to share with our audience?
Yeah, I mean, some of the things that really come to mind when I talk about this are really some reflections. I think the fact that we have this whole issue of biosimilars is just so emblematic of where we've come from in rheumatology in the last few decades. I mean, the fact that we even have a biosimilar transition is because of the success of biologics that were implemented over 20 years ago, and just how much that's done to transform the outcomes for patients. But I also don't want to leave it with the sense that our work is done here. There's still so much unmet need for patients having good control of their disease, for improving their outcomes, for living better, longer lives with all of the different rheumatic diseases that we treat, some of which have good treatments available, some don't have enough, and some don't have any at all. And I think that, we can be encouraged that, we've made this major milestone of introducing biologics and really transforming outcomes for especially patients with rheumatoid arthritis and psoriatic arthritis and so on. But there's still a lot of work to be done through research initiatives. And I think it's going to take, just as it did to get biologics in the first place, patients participating and leading us in research where we need to go. I always say to patients when we start on a new medicine, I'm a better doctor because of the experiences that you share with me as the patient and what you go through with this. We take that back, we take it seriously, and we try to improve things for yourself and for other people going forward. And I know Arthritis Society is doing exactly the same thing. And I think by participating in fundraising and research and promoting the importance of rheumatic diseases to our political representatives and to each other in the community is just so important for us continuing to make gains where there still is unmet need.
I couldn't agree with you more. So let's get into some of the questions that we received tonight. And I will ask this one because it's come up actually a number of times. And I think it relates to my vision of DMODs in the future, disease modifying drugs for osteoarthritis. But the question here that's come up multiple times is, are biosimilars or biologics, can people use those for osteoarthritis?
Well, that is a question that does come up an awful lot. And I think there are a couple things I need to touch on there. First of all, I wish that would be fantastic if we could really move the needle on having treatment solutions for the countless patients who suffer with osteoarthritis, by far the most common type of arthritis. The short answer to the question is no, at least not at this point in time. And that's really important because biosimilars are not changing what the biologics are used for. They're essentially continuing that and really not expanding it in any way. So if it was previously approved for rheumatoid arthritis and for psoriatic arthritis, then the biosimilar version of that medicine will be approved for rheumatoid arthritis and for psoriatic arthritis, and it won't be expanded to other indications. So that's a really important difference between originator biologics and biosimilars. Originator biologics may continue to add more indications as time goes on, and those may be extended to the biosimilars if and when that happens. but we're not using them any differently than the biologic originators were.
Okay. This is a question here from someone who has clearly transitioned from different biologics over time. And the question really relates to, why is it that if you're on one biologic and you transition to another, why would you never go back to that first one that you tried?
Yeah, that's a really good question. And I think there are a couple of ways to think about that. So first of all, in the context of what we're talking about here tonight, which is the biosimilar transitions. So could you go back? And actually, most of the provinces have created the opportunity for people to go back to the biologic originator. That's been built into a lot of those programs if needed. Interestingly, when we followed what's happened in British Columbia and Alberta, many of, in fact, the vast majority of patients have not had to go back to the originator biologic. The vast majority of patients have done very well. Now, it's important to recognize that there is just a background rate of people having flares of their rheumatic disease while taking any biologic medication. And that rate has actually not changed whether you're on a biosimilar or on the originator biologic. It's just that people may end up having a flare no matter what. And if that's the case, then usually you want to move forward. You don't want to go back to what is exactly the same thing. You want to move forward to something that you know is going to give you the best chance possible of controlling the arthritis flare or whatever the disease is that you're treating at that point in time. So that's why you wouldn't necessarily go back. The second way to think about that question is really in the context of access or reimbursement. So let's say that you're taking a biologic medicine that's supported by your private drug insurance plan. In order to go on to the next one, you have to have said, okay, well, this isn't working for me. I've had a flare. I've lost control of the arthritis with the current biologic that I'm on. Therefore, I'm making a transition to the next biologic in the queue. And you work with your rheumatologist to choose what that most appropriate next medication is. So the payer in that case, the private insurance company, will say, well, you didn't do well on that one before, so therefore we're not going to support that one again if you wanted to go back to it. And that's a difficult thing. And that's part of the reason that, we as rheumatologists often will say, well, let's make sure that this is actually a serious flare before we move forward with the next one. We always want to control disease and keep it in remission. That's always the goal. But if this is a minor blip, then we might want to just kind of stay the course so that we don't lose access to that previous medication that's done very well for you for a long time. And really, it becomes a really important discussion between the rheumatologist and the patient every visit, every single time. It's a one-by-one decision. And you have to consider the pros and cons of moving forward, because in some cases, just practically speaking, you can't go back.
So I guess following on from that, as a rheumatologist, if you're looking to prescribe a biosimilar for somebody, and you already showed us that some have multiple biosimilars, how do you pick which one? And does it matter which choice you're making between the biosimilars?
Absolutely. So, we advocate and have always advocated as rheumatologists, as those who are involved in research or those who are not involved in research, we've always advocated for more and more options. This is so key for patients to have options, because no matter what it is, there's no single solution that's going to work for every single person. And there's also no single solution that's going to work forever for everyone. So we need to have options. We need to have choice. And that's part of what I was speaking to, that part of that unmet need before, saying we need to continue to create new options, new solutions for our patients, and advocate for that to be supported. And this is part of the reason that it's great to have so many options available. When we're talking about biosimilars, though, it's almost like too many choices sometimes. You've got so many options available. But I guess I go right back to that key principle that we talked about earlier tonight, that there is no meaningful difference between any of these medicines, the biosimilars, I mean. So if it's adalimumab is adalimumab in terms of how well it works, in terms of how safe it is. So there's really no medical reason to pick one over the other. That doesn't mean that people don't come and have a particular choice that they want to go with. Maybe they know somebody who has been transitioned already and they have had a good outcome and they want to use that one. that can certainly happen. And in that instance, I wouldn't really have any good reason to say go this way or go that way. I think that patient preferences are really important for us to factor in at all times, and including during biosimilar transition. And I guess the other circumstance that comes up from time to time is, which one are we going to have access to? Which one are we actually going to be able to get funded for you? And, we're very lucky here in Ontario. The vast majority of the biosimilars come out, they're all equally funded and listed, and there's really no access reason from a public coverage standpoint to pick one over the other. On the other hand, private drug insurance plans can be quite variable. And it may turn out that, one plan covers options 1, 2, and 4, but not 3 and 5. Or any other combination that way. So sometimes it comes down to those sorts of discussions. But most of the time, I have to say we've been able to sort of pick one and move ahead with it and not look back.
That's a great explanation. Couple questions here. You mentioned that biologics are not used as first line treatment. Sorry, why is that?
Well, I think that there are some really good studies out there that show that could work, and that would certainly be an effective strategy. However, it's not necessarily a cost-effective strategy. And when we think about the first-line medicines that I talked about a bit earlier, so many patients with rheumatoid arthritis will start out taking methotrexate. Many will be on additional conventional anti-rheumatic medicines. We call them DMARDs. Including hydroxychloroquine or sulfasalazine or leflunomide. Those are lower cost medicines because they've been around for longer. They're also small molecules, so they're easier to produce. And from a system standpoint, it makes a lot more sense to start with those medications. And so, before going on to the more expensive biologics, most of the private insurance and public insurance providers will require starting with those conventional anti-rheumatic medicines first. I don't really see that as a major problem. We've had tremendous success with conventional anti-rheumatic medicines for a very long time. And in fact, in the 1980s, when methotrexate came out, this was a complete game changer for people with rheumatic diseases, especially rheumatoid arthritis, but many others. And methotrexate continues to be the cornerstone of treatment for rheumatic diseases around the world, just as it is here in Canada, even to this day. And many patients do not need to go on to advanced treatment with biologics. So, I think the short answer is it's more cost effective, but in many cases it's just not needed. So, you need to figure out who, and this is the great art of medicine, as it were, is finding out who needs to be on certain therapies and finding the best combination of medicines possible for this patient, which may be very different for another patient.
Yeah, so picking up on that cost effectiveness, could you just give people a sense of, ballpark, what are we saving per patient, say, when you're comparing an original biologic to its biosimilar?
Well, what we're saving is approximately 30% of the total cost of a originator biologic. And the reason I'm pulling that number out of what seems like thin air is because that's what's been essentially sent to us by national and provincial payers who have negotiated the prices for biosimilar medications in Canada. But that's very much an average. And, it's going to vary quite a bit from disease to disease, from medication to medication, and from province to province. But that's really important to just recognize that there is a substantial cost savings, but these are still not inexpensive medications. So we still are not, just sort of transitioning and putting everybody on these medications because it would be very costly to do that. But certainly if there's a medical reason that somebody needs to be on a biologic medicine, we are so blessed to have access to public coverage in Ontario here, where I practice, in all of the jurisdictions across Canada. People can get access to their medication, which is a real blessing for us here.
So you mentioned within the slides, batch to batch variability. And then there's a question here about, is it possible for one injection of biosimilar or a biologic original biologic to work and have subsequent injections not work? And the question relates to, is there something happening with respect to different batches that people are receiving?
Yeah, that's such an interesting question. And, I've certainly seen that clinically, anecdotally speaking, and I've certainly heard patients talk about that. My own patients, other people who have experienced this and certainly work with patient advocacy groups and that sort of thing. This is not a common occurrence. So it's by far not the majority. But I think that what most of the studies will tell us is that there are normal ups and downs of rheumatic disease. What do I mean by that? Well, rheumatic diseases, autoimmune diseases are essentially a permanent disease. We don't cure anything in rheumatology. I wish I could say that we did. But what we do is we help to keep the inflammatory disease in remission. But there are times throughout somebody's life when the disease will wake up a bit. Sometimes it's a big flare, sometimes it's a small blip, as we would call it, so a small increase in disease activity. But there are ups and downs. And it's entirely possible that those background ups and downs are what we're experiencing when we think there may be sort of changes from dose to dose. However, people have really said, well, when I had this batch, I was doing really well. When I had the next batch, I didn't feel like it was working quite as well. And then I switched to the next one after that and things kind of got better again. Is that due to the medication? There's no clear evidence that that is the case, but certainly it is something that people report. What I usually say is, if there's actually an issue with this, let's see, okay, can we switch the batches here? Meaning, can we get a different lot number and see if it is the medication? If that's still not working, then that probably means that this drug is not working for you anymore, and then we should move on to a different one.
So that was a great segue into my next question, actually, around how do you know when a biologic is working? Is there a way to test it, through blood work, or are you just going by people's symptoms?
Great. So it's kind of actually all of the above and more. So in rheumatology, we're very much focused on the physical examination, and this is part of the reason that it's been very challenging for the last couple of years to provide the best care possible through virtual care. I think we've done a very good job in most cases of adapting to virtual care, but at least when it comes to assessing how well a medication is working, we need to be able to examine the patient. We need to be able to see how much swelling there is, to know whether there are 2 swollen joints, 5 swollen joints, 10 swollen joints, and whether that's improved from 2 or 3 months ago when a change in medication was made. We also measure things like symptoms, which you mentioned, and our patients report those on different scales, and they also just come in and say, hey, Tom, you know what? I'm feeling a lot worse. Like, we need to talk about this. We need to figure what's going on. And sometimes that's because the disease has flared, and sometimes that's because of something else that's actually going on. And then we do measure blood work as well. And, those would be done either every month or every 3 months or some interval there. But we're following those more or less objective measures in terms of blood work. When we're taking all of those measures and putting them together into either a disease activity score or some other way of measuring how active the disease is. So that's how we objectively assess whether a medication is working. And then we continue to monitor that over time. So, I often will say, in the beginning, we're going to be seeing each other a lot more often until we get this disease under control, meaning in remission. And then, I'll stop bringing you into the office so frequently, and maybe it'll be every 6 months or maybe even once a year. And, you'll forget what I look like, which will be nice for you. But that is a really good sign that things are going well when we're able to space out visits a lot more. And that's the goal, to live life with arthritis and have the best quality of life possible with the disease under good control.
What about side effects? Are there any different side effects for people to worry about related to biosimilars versus original biologics?
Yeah, this one is, I think, really what we kind of covered off earlier, which is, when these medications are studied, are they demonstrating the same safety and efficacy as the originator biologics? So The answer to that is no, there's no clinically meaningful difference in terms of how safe they are. There's no difference in terms of which types of safety issues could arise. But with any biologic medicine, there are potential safety issues that should be discussed. And so you will always go through that with your rheumatologist or whoever's providing you the biologic. You'll talk about serious infection risks. You'll talk about, heart failure. You'll talk about things like tuberculosis screening and all these sorts of things that happen before we start taking a biologic medication for the first time, and then periodically checking in over the years that come. But there's really no difference in terms of whether it's a biosimilar or an originator biologic.
And what about the original clinical trials that would have been foundational when the biosimilars were being approved? A few questions here about would they have been similar to the clinical trials that the original biologics would have gone through? Again, I guess just underlining these questions around, are there differences that people should be worried about?
Yeah, absolutely. So again, there really aren't any meaningful differences that people should be worried about. And I think that's the take home message. Study after study, and like I said, over 170 now worldwide that have been done across many different diseases by many different groups, rheumatology, dermatology, gastroenterology, and so on. And even outside of the inflammation realm, biologics have been compared to one another. And so, in a good way, they're some of the most boring results to review in clinical trials because they're exactly the same. They follow the same curves. People improve at the same rates over time. You have exactly the same types of safety signals that come up, fortunately, which are very uncommon. But it doesn't really matter whether it's for rheumatoid arthritis or psoriatic arthritis or any other indication. The biosimilars continue to just be exactly the same as the originator that they're compared to. I think what gives me the greatest assurance as a rheumatologist and somebody who actually reviews the studies is that we're actually comparing these head to head. So that means we're taking the original version, the originator biologic, and randomizing patients to that particular originator or to the biosimilar and watching them head to head compared over time to see how well they do. That's the gold standard. That's the randomized clinical trial way of assessing things is the gold standard to know whether things really are similar or different from one another. So that's why I can say, if we were going to identify differences, I feel like after 170 plus studies, we would have identified it by now. So we can be quite reassured about that.
Great. Maybe you could just touch on for a second about how these biologics are working. We have a couple questions here about, are they suppressing the immune system? Are they actually changing the disease, or are they just masking pain? So could you just spend maybe a minute about how these work in your body?
For sure. So the concept of disease modification is what we really rely on in rheumatology. And what does that mean? Well, covering up symptoms would suggest that you're not changing the disease at all. Right. So if you're just treating the symptoms, then potentially the disease is carrying on and causing more damage, causing other complications and that sort of thing. That's not what biologics are designed to do, nor are other conventional anti-rheumatic drugs or DMARDs. DMARD, by the way, DMARD stands for disease modifying anti-rheumatic drug. It's the disease modifying part that matters. So in order for a medication to be approved as disease modifying, it actually has to show that it has the ability to suppress inflammation and to prevent damage to a joint. So let's say if it's rheumatoid arthritis, it has to actually prevent erosions from happening and damage to that joint. That's how it gets determined as being disease modifying. And that's the main reason that we continue to follow whether somebody is having active swelling in their joints or not. If they're having 2 or 3 or 4 swollen joints, well, that disease isn't as well controlled as it could be. Certainly better than if it's 10 or 12 swollen joints, but still not as good as it could be. So that's what we're really trying to treat, is suppressing inflammation so that it can't cause damage, can't cause long-term complications from the disease. The biologics do that by targeting inflammatory proteins that drive that inflammation. So we're really suppressing that inflammation. So hitting that target to suppress the inflammation and change how active that disease is so that it can't cause the same sort of damage. The only thing that would be better than that, as I mentioned earlier, would be a cure, meaning you take the medicine once or twice or for 5 weeks and then boom, it's gone and it never comes back. That's not where we are in rheumatology. We are at the point of suppressing the disease and keeping it in remission. That's the target. But for the vast majority of people, that means continuing to take these medications long term. But that's the safest thing to do because uncontrolled inflammation can cause serious damage, shorten lifespan, poor quality of life, and that's just not acceptable.
Maybe just a few quick questions here before we have to go. The first one, what about biologics or biosimilars for pregnant women? Any concerns?
So again, this is where you can say that, what would have applied for biologics would apply exactly the same for biosimilars. Now, it's important to point out that biosimilars have not been tested in pregnancy studies to the same way. There are pregnancy trials that have been done with biosimilars, and again, they show exactly the same results as the biologic originator. So no reason to suspect that there would be an issue that way. But obviously, pregnancy clinical trials are a lot fewer than they would be from drugs that have been around for a lot longer. However, we are clearly at a point now where there are important considerations for some biologics that more so than with other ones. And when it comes to TNF inhibitor biologics, the majority of those are considered to be quite safe in pregnancy. There are times when you need to interrupt it, certain times during the pregnancy, and this should always be discussed ideally before pregnancy is contemplated. But it happens where you have to address it after pregnancy is already initiated. But there are certainly times when you want to interrupt it. The most important thing, though, is controlling the disease during pregnancy, because that can have a major impact on the mother and the baby. So we have to weigh those different priorities. In terms of the safety, though, it's exactly the same as it would be for the originator biologic, and that's what your rheumatologist would discuss with you.
So would you essentially say that biosimilars are generics?
Would I say that they're generics? I've asked that question before. What really is the difference here? I figured this would come up. And I did touch on it briefly at the beginning. So generic is a term that is used to talk about small molecules. So I mentioned aspirin. Pretty much any other medication that you can think of that's taken in a pill form is considered a small molecule. And if it doesn't yet have a generic, it will have at some point in the future. Generics is a term that's used to describe the carbon copy, the identical replication of a chemical medicine, small molecule medicine. We can't use that term when it comes to biologics because you can't do a carbon copy of a biological thing, right? Even identical twins in the same family have minor differences that could be detected either with certain types of blood testing, certain things. So at the end of the day, they're so similar to one another that it doesn't have any difference in terms of how well it works in clinical practice, how well it controls the disease, and how safe it is. But at a chemical level, you can detect minor differences. Is that important? Clinically, it's not important. Otherwise, we'd be much more concerned about this. We're not concerned about it at all. But at the same time, we can't use the same term generic. So we use the term biosimilar because they're so close to each other. We might as well call it bioidentical, but that's harder to say.
So I don't know if it's fair to ask you this with 1 minute left, but what are some exciting new disease-modifying drugs, biologics or otherwise, on the horizon?
Some new ones on the horizon. Well, there is always a lot of activity going on in rheumatology. Not enough, in my opinion. I think that we have so many unmet needs that there needs to be more. There are more medications coming for more diseases that don't yet have a lot of treatments available. And I think the top one that comes to mind is for lupus, in particular lupus nephritis. So that's when your kidney becomes inflamed and you can get kidney damage from lupus. There are new biologics that are either approved or shortly expected to be approved for treatment of lupus nephritis and for lupus in general. That is very exciting for us in rheumatology. Lupus is fortunately a very rare disease, but for those that have lupus, is a very serious disease and causes lots of long-term complications, in particular due to the fact that prednisone is needed, so lots of steroids, and we need to find a way to replace those steroids with something that's safer and more effective. And so this is an exciting time for lupus in particular. There are other things that are being studied for vasculitis, which is also a rare type of autoimmune disease that are in the pipeline, and I'm excited to see where that goes. Not quite as far along as it is for lupus, but I think some of our diseases that we've really struggled to treat with, sort of anything but prednisone for, throughout many decades, we're starting to see new and exciting opportunities come along. There are others, but you only gave me a minute, Sean, so I'll stop there.
I'm very sorry, but thank you so much, Tom, for sharing all of your knowledge with us today. That was just fabulous. We would like to take a few moments to get the audience's feedback if we could on today's presentation. So those of you watching the Zoom, you'll see 2 poll questions come up on your screen and answer options. So just do click on the response that reflects your thoughts. Simply, have you talked with your healthcare provider about transitioning to a biosimilar? And importantly, did you find the information in today's webinar helpful to you? We will also be sending out an evaluation form when we email the recording. So if you were unable to access the poll questions, you'll definitely have the opportunity to give us some feedback at that time. And we really do use the survey feedback to shape our Arthritis Talks webinars, so we truly value your input. Once again, we are grateful to our partners, Pfizer and United Way Winnipeg, for their support of this event. And before we wrap up tonight's session, I wanted to take a moment to tell you about Arthritis Connections, a new program we are launching. Arthritis Connections are virtual workshops that enable people living with arthritis to come together, learn about their disease, and build relationships with one another in similar circumstances. Our first session will be on March 29th. We will be sending you more information on how to register an email you'll get tomorrow with the recording of this session. So this concludes Arthritis Talks, Understanding Biosimilars. On behalf of all of us at the Arthritis Society, thank you for joining us tonight.